Everyone, and welcome to today's APS seminar. Before we begin, I'd like to remind you to please enter any questions you have using the chat or Q&A feature, and they will be answered at the end of the session. It's my pleasure to announce Dorothy Sweet as today's speaker. So Dorothy is originally from Wesley, Iowa, and she grew up on a small family farm outside of town. She attended St. Mary's University of Minnesota and graduated in 2019. She also completed a 9-month research internship with Corteva Agriculture. Dorothy is passionate about making life easier for farmers, and when not working, she enjoys hiking, camping, knitting, and spending time with her family. Thank you, Dorothy.
Thank you, Catherine, for that nice introduction, and thank you everyone for coming. As Catherine said, my name is Dorothy Sweet, and today I'll be talking about the use of high throughput phenotyping tools for evaluating the interaction between plants and their environment. So first off today, we are going to talk a little about current and emerging challenges in agriculture, why we care about corn, the phenotyping bottleneck, and then the 2 objectives of my project and the summary. So first off, we're going to talk about agricultural challenges. So the first of the agricultural challenges is population growth. As we all know, there's an increasing population. As we can see right now, there's about 7 billion people on the planet, but it's projected that by 2100, we'll have around 11 billion people on the planet, which is causing an increasing demand for food while also decreasing the amount of land that we can grow this food on. We're also struggling with climate change. A warming world means more extreme weather events, and we need to learn how to cope with those weather events. There are also a change in agricultural inputs. There are decreasing resources such as soil nutrition and water, but there's also a decreasing number of farmers to take care of fields. And lastly, we have challenges with societal inputs. There are changing tastes, expectations, and an overall distrust of the agricultural community sometimes. So we will be focusing on these 2 challenges of climate change and agricultural inputs for my project. So now that we've talked about climate change, I want to talk about how that is leading to changes in crops. So flooding can drown out crops and can also remove fertile topsoil from the field. Excessive precipitation can lead to that flooding and can prevent farmers from getting their crops in on time. So excessive precipitation after the crop is already in can also lead to damage to the existing crops. The crops that are growing in standing water can suffer from root death or inhibited photosynthesis. Additionally, this excessive water can lead to an increased fungal and disease pressure. Warming climates can also allow existing pests and diseases to survive in new locations that they previously would not have been able to survive in. Drought can also cause a loss of arable land and crop death. However, farmers may try irrigation on land that's never been irrigated before to try to prevent a total loss of crop. But heavy irrigating can quickly drain remaining surface water sources and may make significant dents in aquifer contents, which may lead to water and other natural resources that will be lost. And that will in turn remove habitats and food for beneficial insects. And driving many aspects of this change is the excessive heat. In addition to all of the issues we already talked about, heat can reduce existing surface water, disrupt flowering and pollination cycles that have already been determined, and increase weed pressures. So for these reasons, we need to be able to breed plants that can survive and thrive in these new and challenging conditions. But why should we worry about corn specifically? So while yield has steadily increased from 1960 to 2010 for corn, with a few setbacks due to weather events, those positive gains may not continue as the areas marked unusual climate events, they become more common. So this figure summarizes studies that predicted the change in corn yield over the next 100 years. Some studies still estimate a positive yield in the next 100 years, but nearly half of the projections beyond 2050 indicate yield decreases greater than 10% and only getting worse from there. So we care a lot about corn, mostly because there are many uses for it. We depend on corn for animal feed products, ethanol, exports, and even a small amount of food for ourselves. So 1 bushel of corn can create many pounds of other products such as corn starch, corn syrup, sustainable plastics, and ethanol, along with all of ethanol's useful byproducts. In addition to all of its uses, corn is economically important at a corn crop value of $52.7 billion, which accounts for 25.7% of all national crop value in the United States in 2019. So as we've already discussed, corn yield has been increasing quite a bit since people started recording the average yield in 1866. From 1866 to the late 1930s, which are the yellow dots on this graph, very little change in average yield occurred. And then in the 1940s and 1950s, which are shown by the red dots, they saw increase in yield mostly due to improvements in germplasm and the use of double-cross hybrids instead of open pollination, which was an increase of about 0.8 bushels per acre per year. Then from the mid-1950s on, which are represented by the blue dots here, the yield gains doubled to an average increase of 1.9 bushels per acre per year. And this large increase was caused by genetic improvements and increased adoption of modern management practices such as nitrogen fertilizer, chemical pesticides, and advances in machinery. So even with all of these improvements, there is a lot of variability in corn yield across the United States. And in this map, the darker colors show higher average yields in those counties. But we're in Minnesota, so let's focus on just Minnesota. As you can see, there's still quite a bit of variability in Minnesota as well, below county, below 150 bushels per acre, and the high of 185 plus bushels per acre. And most of the best yielding counties are in the southeast portion of the state, as we would expect. Now, the average yield for 2019 was 174 bushels per acre. You can see that some counties in northern Iowa here yielded more than 200 bushels per acre on average, but that's nothing compared to the max yield from the National Corn Yield Contest of 616 bushels per acre. There's a huge gap between these two numbers. That's 442 bushels per acre. But to be fair, a large contribution to this gap is an extremely unsustainable amount of maintenance and resource inputs that were needed to achieve this 616 bushels per acre. But even without the huge increases in maintenance over the years that are largely contributed to the increase in yield, the germplasm that breeders are forming are also contributing greatly to the changes in yield over the years. A study in 2014 grew historical hybrids from the mid-1930s through 2014 in identical growing conditions, but at their preferred planting density. This figure from the study shows how much of the increase in yield is still due to genetics. And for reference, 12,533 kilograms per hectare is approximately 200 bushels per acre. So that's about like the top of this graph. So now let's talk a little bit about the pipeline that breeders use to make those improvements. So this is a very simple diagram of the plant breeding system. This first triangle on the left represents the genetic variation present and the number of entries. And this triangle on the right represents the stringency of testing, or the number of locations that all of those entries are tested at. As a beginning to the path for breeding selection, many inbred lines are created from the inbreeding of offspring from 2 inbred parents. Double haploids are more common now in the corn breeding pipeline, but I'm not going to cover double haploids because they're complex for the sake of time. And this group of inbred lines decreases in the number as the stringency of testing increases. So first selections are often made using marker-based selection to get rid of the individuals who might not have certain desirable characteristics such as disease resistance. Then notes are taken on the phenotypes of the inbred's ability to cross with other inbreds to create hybrids, which is called test crossing, and eventually hybrids that are product potentials. And eventually, after all of this work, which takes around 10 years, a new hybrid is deployed, which is the product that is sold to farmers and that you can see out in most fields. So now let's talk about some of the important phenotypes at each of these stages. Each stage has some unique phenotypes. First, we're going to talk about the phenotypes for inbred. So each of these phenotypes is collected from a different part of the inbred plant. And I'm not going to be going over the reasoning for why we collect these phenotypes, but if you'd like to know, I can cover that in questions later. So yield is determined, and it's also somewhat linked to ear size. In addition, we also have ear height and flowering date, plant height and disease susceptibility, tassel size, tassel extension, sterile tip, and pollen count. Next, we're going to focus on phenotypes that breeders are interested in for hybrids before those hybrids are deployed. These phenotypes are very similar to those for the inbred plants, except that they are on hybrid plants. So yield is more important at this stage of testing, and grain moisture and test weight are also important due to their influence on yield and grain quality standards that producers have. In addition, we have ear height, plant height, and disease susceptibility, and lastly, lodging and green snap. Lastly, we're going to be focusing on the germplasm that is deployed and which traits farmers actually care about. So these first 5 traits, grain moisture, test weight, lodging, green snap, and disease susceptibility, are very similar to the last category, so I'm not going to be covering them again. But in addition to this, farmers care about stay green, the maturity zone that those crops are grown in, and lastly, we've made a full circle back to yield. And this is most important to farmers. While breeders work on the genetics of a plant that will yield well, farmers concentrate more on providing the best and most economic growing conditions to provide a good yield and good income for their farm. So after looking at all of the time points separately, we can see that there are a lot of phenotypes that are collected throughout the corn pipeline. Collecting all of these traits can be very time-consuming. For this project, we are going to be focusing on yield and plant height for all stages of testing and adding 2 new traits, growth rate and canopy closure. These traits will really give us the response to the environment that we need for combating climate change. So as plant height is extracted for each time point, a growth rate can show the plant's change in plant height over time, which may give us more data about each genotype's response to environmental factors. Instead of only getting a static view of the plant's response with plant height, which we would get at the end here, growth rate allows a more complete understanding at many time points. And by implementing growth rate and canopy closure, we can get a better idea of how these genotypes would respond to climate change. For instance, if a rainstorm were to occur on this date after a time of drought occurring here, we can see that these 2 genotypes responded differently to it. Until that point, both genotypes had responded similarly to their environment. After the rainstorm, however, genotype 2 quickly takes a sharp advantage of the newly beneficial conditions, and genotype 1 doesn't take off as fast. So this could be very beneficial in identifying which genotype is more suited to surviving difficult conditions. But we would need to collect this plant height many times throughout the season to attain this growth rate, which, as we said, is very time consuming, especially when using traditional phenotyping methods, which would, or tools, which would be like a pen and a paper and a person and a stick in a field. So what types of tools are available to potentially speed up this process? So there are a lot of different levels of phenotyping platforms available, and we're going to be covering a lot of the outdoor platforms from broadest area covered to narrowest area covered. And there are some satellites that are constantly orbiting and taking pictures of the Earth. These satellites have a variety of areas that they cover in one image. Some cover thousands of square miles with one image, while others cover much smaller areas at a time. And satellite imagery has been used in the past to study things like ground cover, and we can access data from some of them for free or for a fee. Unmanned aerial vehicles, or UAVs, are air vehicles without pilots on board that are part of an unmanned aerial system, which includes the UAV, a ground-based controller, and a method of communication between the two. A boom-mounted sensor involves mounting a sensor, such as a camera, on the boom of a high-clearance field vehicle. This vehicle then drives through the field collecting data. A rover is very similar to a boom-mounted sensor in the sense that it's still a ground-based vehicle with a sensor mounted to it to collect data, but these are usually remotely controlled. A rail-based gantry system is a permanent location sensing system that has rails enclosing a small area of land, such as 2 or 3 acres. And a large sensor system can move back and forth over the area and collect data automatically. Then handheld plant sensors can collect data similar to those other systems, but usually only on a single leaf at a time, which can be very time consuming. And lastly, cellular phenotyping collects data on a cellular level, which greatly decreases the amount of different data that can be covered. So now that we've covered all of those potential phenotyping platforms, there are a few things that we need to keep in mind when deciding which ones would work the best for my project. First, we need to consider what kind of spatial resolution we need. So spatial resolution tells us the size of the pixels we attain and how much detail is in each full image. And as you can see in this diagram, a pixel size of 30 meters by 30 meters is so large and such a low resolution that we can't even tell that there's a structure inside this pixel. It's just blue. And as the resolution increases and the pixel size decreases, we get more detail and can determine that this might be a house that we're taking an image of instead of maybe like a parking lot. So for this project, we'll be needing a fairly high resolution as we're trying to capture plant height variability throughout the field. Secondly, we also need to consider temporal resolution, which is how often can we revisit the area of interest and collect data, and how long will it take to do so. With old methods of collecting traits like plant height, there was very low temporal resolution. It takes a lot of time and people to collect plant height at the end of the season, let alone multiple times throughout the season. But if we want to collect things like growth rate, we need to have a higher temporal resolution. So when we're considering those possible imaging platforms for this project, we need to keep these spatial and temporal resolutions in mind. So based on those, we decided on UAVs for this project. So now that we've decided on a phenotyping platform, let's go over some of the pros and cons of that decision. Aside from all of the downfalls of the other options, UAVs may have many benefits that are associated with them. UAVs are relatively cheap. For instance, one like this could be purchased for around $1,500. Secondly, UAVs are attainable. It's fairly easy to get a license for drone piloting. They can also collect our data very quickly. This flight pictured here on the monitor is one of my regular flights from this last summer, which covered over 2 acres of plots with less than 20 minutes of flight time, which is much faster than collecting plant height by hand. And then also UAVs are versatile. They can also collect data where you want and in many conditions. And as you can see, this drone is being flown over a field of wind-damaged corn, which would be impossible for the ground-based sensors, such as the boom-mounted sensor, as they would run over all of the corn. Lastly, UAVs are flexible. They can collect data almost anytime you want, and they are very easy to take with you from place to place. So now that we've covered many of the benefits, there are still some drawbacks to using them. And we're going to be covering 4 of those issues with a little more detail on 2 of them that are more relevant to my work. For instance, there are some restrictions on how far and how long you can fly a drone. This is due to low payloads, including battery and sensor weight, as well as government restrictions on how far away and how high drones can be operated. Improvements are constantly being made in these drone payload capabilities, but biologists aren't really working on them. There's the second issue of data size. Many of the images collected by a drone have a very high resolution and therefore take up more storage space than an average picture you would take on your iPhone, around 8.6 megabytes each. So one flight on one field, after all the processing, can take up more than 20 gigabytes of storage space. And this isn't a whole lot, especially if you compare it to like hyperspectral images, But as temporal resolution increases, so will the overall amount of data that needs to be stored. And this is another area in which improvements are constantly being made by non-biologists, but mostly for other purposes. So for these next upcoming two issues, I'm going to be going a little more into detail about how they're being handled. So this first of those two issues is determining where the ground is in comparison to the tops of the plants. Now, as you can see in this picture, very little ground can be seen in between the rows of the plants. So it's very difficult to determine how tall the plant actually is, especially if that field is planted on uneven ground. And I've read some research that encapsulates 4 different ways this can be calculated accurately, but I'm just going to be going over 2 of the ways that are relevant to my research. So the first of these 2 methods is the different space method, which involves collecting a flight either before the crop comes up or after the crop is harvested. This flight is called the ground flight and serves as all the ground pixels for your data flights. A script is written to subtract those ground pixel values from the pixel values of the data flight after other processing is finished. Exposed alley subtraction is another method which was developed by Sarah Tirado while she was here at the U. But this method only works for plots in which there are alleys surrounding them. The alleys can be used or can be identified with classification systems, and then the pixel values of those alleys will serve as the ground value for all of the bordering plots. So now that we've covered how ground is being handled, the last issue that I'm going to cover is processing. It's important to get accurate data from processing the images, and sometimes this can be difficult due to the complexity, the accuracy of the data, or the processing time. So many groups are working on this, but each group has its own benefits and downfalls. And today I'll be talking about the pipeline I use, which was also developed by Sarah Tirado while she was here at the U. And while this pipeline works fairly well, it uses 3 different softwares, Agisoft, Metashape Pro, QGIS, and MATLAB, and it's pretty time consuming. If we're going to be increasing the temporal resolution to a very high rate, the processing timeline will probably need to get faster. And many of these groups are developing faster and simpler programs with little effects on the data quality. So now that I've covered all of the pros and cons and why I'm using UAVs, let's circle back to the breeding pipeline and phenotyping to discuss how UAVs will be used specifically in my project. So for my project, I will be focusing on specific parts of the breeding program. So for objective 1, I'll be focusing on the part of the pipeline which is highlighted here. In this box, there are many variable individuals tested in some environments over a few years. And in order to replicate this scenario, I will be looking at many genotypes grown over a 3-year period. So for this first objective, there are 3 questions that I am trying to answer. Does variability and growth rate of different genotypes stay consistent across multiple environments? Can yield be predicted accurately using a model with plant height, growth rate, and/or canopy coverage? And lastly, can we map the genetic basis of the genotype's response to the environment? To answer all of these questions, I'm gathering data from a subset of the Wisconsin Diversity Panel, which is being grown on flat and plotted land similar to that of a breeding program. The full diversity panel shown here is composed of 942 diverse inbred lines with some shared ancestry from 11 different subpopulations. And for this project, I am using a subset of 500 inbred lines that were picked based on a 2-week flowering window. And this panel is very ideal for my research due to all of the other research that has already been done on it and all the available data for it from these papers. So in addition to all of that data from those papers, we have collected flight data over many years, as well as manual measurements. Although this is the first year, 2020, that we also have yield. Additionally, 2020 is unique for having 2 replications at 2 different locations, here in St. Paul and in Waseca, Minnesota, as opposed to just St. Paul in previous years. So with this flight data from multiple environments over multiple years and the yield data from this year, we determine how growth and the yield for the diverse genotypes in our panel are affected by the environments that they are placed in. In order to evaluate the environment's effect on the genotypes, we first need to extract plant height and and phenotype from the field. This starts by aligning all the photos to one another. So this is one example image from a flight. In order to align these images, the software identifies individual points in between the images that are similar. And this process is simplified for the software by using ground control points or consistently marked locations called GCPs and identifying them for the software. So I enlarged this GCP to make it easier for you to see. So from these points of similarity, the software then aligns the photos to one another. And in this image, every black line signifies a picture that has been properly aligned to the pictures surrounding it. So from these aligned photos, a dense point cloud, which is a 3D image of the field, an orthomosaic, which is a compiled aerial image of the field, and a digital elevation model, which shows elevation differences, can be formed. From these data, each single plot can be identified by a bounding box and extracted, and then plant height and canopy coverage can be pulled from that image. So we know that we can trust this data due to the high correlation between this extracted plant height and the manual measurements that I completed on each flight day. So after confirming that we have reliable data from the drone, we can create a growth rate model like this one. And in turn, we can determine whether or not these growth rates are predictable in other environments based on the locations and years of data that we have. So now we have some initial data from some of the flights I took this summer, which show the height data across all of the days until terminal height has been reached. There's quite a lot going on in this initial graph, so I'm going to be breaking it down a little bit. So this circled area here shows the data from the first few flights. As we would expect to see, there's a general upwards trend in the height of the plants in the plot. However, if we look at each of these days separately, we can see that there's a lot of variability of height within plant days, including plants that are a lot taller and some plants that just never seem to grow. And these plants that probably never grow are most expected are probably from empty plots that I haven't removed from the data yet. Additionally, we can see that there's a large difference in some of the growth rates, which is what we're most interested in, as this growth rate is a lot steeper than that growth rate. Lastly, on this graph, I want to point out one more area of interest. This circled area shows quite a few plots which are getting shorter from one day to the next without any large wind events in between to knock down any of them, which doesn't really make any sense. So this data confused me until I remembered this day on the right, where our lab went out and killed all of the weeds that were growing in the alleys. And since I was using the exposed alley subtraction method, the weeds in the alley became the height used by that method for the ground, which made the corn plants around them look shorter. And this error was easily able to be fixed by calculating height through the difference-based method instead of the exposed alley method. And this simply illustrates that the additional data quality control is necessary even if the extracted heights correlate well to the hand height measurements. So in addition to all of this, previous research has shown that plant height correlates well with end of season yield. However, we can see from this figure here that the stage at which plant height is collected can affect how well it correlates to yield. In addition to that, we can see that the best correlation to yield is from plants which are already at the V12 stage. And in this image down here, which shows a V9 plant and a VT plant, with V12 being in between them, we can see that a V12 plant is very tall, too tall to make any mid-season management decisions. So growth rate may be able to fix this issue. As we already determined, there's early variability in these growth rates of all these plots. And we hypothesize that the data from multiple days together will be able to better correlate to yield than any single date did in that previous plot. In order to test that hypothesis, we need yield. So this fall, we hand harvested, shelled, and collected data on each plot to calculate the yield. And this data will eventually be used to determine if there are useful connections between yield and early season growth rate. But can we map the genetic basis of the genotype's response to the environment? So how do we go about answering this question? So considering all of the data that's already been collected on the Wisconsin Diversity Panel in those previous research papers I talked about, and all of the data I've collected myself, a genome-line association study, or GWAS, seems like the best option. But what is that? So GWAS is an observational study that determines if there's a connection between genetic variation and phenotypic variation and how they relate to each other. So I'm going to be using the example of plant height to explain it. We have a large group of corn plants here, and some are tall and some are short. So now we are going to be taking all of these corn plants and dividing them by phenotypes into short and tall groups. So now that we have those plants divided by their phenotype, we're going to genotype them. And then we are going to identify the consistent differences in genotype between the two phenotypes. These differences in genotype seem to have a connection to plant height in this example. And after identifying those points of differences across the genome, I will confirm the gene's connection to the trait. And this process will be fairly simple because of all the data I've collected and the data we already have from previous research. Additionally, I will not only be able to look at the genetic conditions to height throughout time, but I will also be able to look at the changes in growth rate over time. From this information, I will be able to determine if the genes stay consistent from one time point to the next, and whether or not the genes regulating the growth stay the same. So after discussing the first objective, which has variable genetic material in flat land, we're now going to be switching gears a little bit and come back to see where in the breeding pipeline I'll be focusing for my second objective. So the second part of my project will be focusing on this part of the pipeline. At this stage, there is little to no genetic diversity, as you can see by this triangle here. And most farmers plant only one or two genotypes in a part of the field. And these genotypes are also going to be grown over a much wider variety of locations, which include fields with variable elevations and soil types. So this is a basic farmer's production field. So you can see there are some areas with maybe some standing water that caused a decrease in stand or something like that, which is pretty typical. And from that field, we can get a yield map from the combine, which only helps with postseason changes for the next year. But what if we want to make changes in the current growing season have impact on our current yield? Well, a farmer can have an agronomist come out frequently, or they can scout the field themselves, like quite frequently, which is time consuming. It's also expensive to have an agronomist. And if you're a busy farmer, your time is your money. So scouting your field very frequently is also expensive. So we need a quick and accurate assessment for targeted management, and that's where this project comes in. So in this objective, we are also trying to answer 3 questions. Can accurate data be extracted from flights over uneven ground? Can crop models from the previous objective be applied in this environment with higher variability? And can accurate predictions of yield be predicted early enough to make mid-season management decisions and impact the current crop? So the first challenge of this objective is whether or not accurate data can be extracted on a heterogeneous landscape using the difference-based method. In order to test this, I completed multiple flights over the field, which included a ground flight that you can see here. We also randomly picked 40 plots out of 10 foot long throughout the field. During each flight, manual measurements were taken in each of these plots. After extracting that data, you can see that there's a very strong correlation between the extracted heights on the bottom here and the manual measurements that I took on the left. And this data looks very promising. So how much of this data do we have? If you recall this flight frequency diagram from before, for this objective, I only needed to complete 10 flights due to the hybrid reaching terminal height before the variable inbreds did that I took 11 flights for. And this first flight here was a ground flight that was taken before the crop came out of the ground. Since we determined in that previous graph that the data is trustworthy and that since there aren't conventional plots across the field, we'll be able to get a map of plant height that looks something like this for each day, which covers the entire field, with the shortest plants being red and the tallest plants being blue. So then we also hand harvested the 40 plots we found formed in this field. The corn from these plots was shelled, weighed, and measured for test weight and moisture in order to calculate yield. No other data was collected from these plots, as we do not intend to determine what caused the variation in growth rates, only what growth rates can tell us about yield. So the plots that were hand harvested were done as a cautionary measure. As you can see on the left here, we at least have yield data for those 40 plots, which are fairly variable. And hopefully we'll be getting a yield map such as this from the combine when the rest of it's harvested. And since something could go wrong with a combine yield map, that's why we chose to harvest the 40 plots by hand. So at least we would get some data from the field. So now the differences in these growth rate graphs, such as this one, can be connected to the yield data that we have. And the area in the red circles shows some of the differences in the growth rate between these 2 days. This one has a much higher, faster growth rate than some of these do that are already taller. And this may give us evidence for the yield, which will in turn give us targeted data for mid-season management opportunities in the future. This data will not be giving farmers information about what is causing the variation they're seeing. Instead, it will identify low yielding areas early in the season before you get yield, where there's still something that can be done about it. And this will help farmers identify problem areas that may not be obvious with only scouting, or that will need to be evaluated for issues such as nutrient deficiency, and that may also reduce over applying nutrients like nitrogen. So in summary, after covering all of this material, I want to remind you all about some key takeaways from my talk today. The climate in which we are trying to grow our crops is constantly changing. In order to continue growing crops like corn, breeders are constantly making improvements to the germplasm being grown. But phenotyping speed is bottlenecking the amount and kind of data that they have available when they're making their advancement decisions. So UAVs are being used to improve the phenotyping process. These genotypes respond differently to the environment, which can be linked to their underlying genetic traits to help make predictions earlier in the season. These traits and underlying genes can in turn be connected to yield data, which can be used to develop an easy and affordable method to identify targets for mid-season management. I would like to thank my advisors, Dr. Corey Hirsch and Dr. Candace Hirsch, and everyone in Dr. Candace Hirsch's lab. I would also like to thank Sarah Tirado, who did a lot of work on this before I got this project. And I would also like to thank all of the people, mostly in our lab, but some from Nathan Springer's lab as well, who helped collect all of my corn this fall. And then I would also, of course, like to thank my funding, which is from Minnesota Corn Research and Promotion Council. Are there any questions?
That was great, Dorothy. Thank you. So now, once again, anyone who has any questions, please type them into the chat function or the Q&A function, and Dorothy will be happy to answer them. Just give people a minute to type here, Dorothy.
While we're waiting on that, Dorothy, this is Don.
Mm-hmm.
So when you were going out and you were indicating that you were not trying to figure out what was causing the difference in the growth rate, but rather just indicating that it was, but you were indicating that by making this early assessment, you might be able to do something in the field. If you don't know the cause, how would you go about correcting the problem? Could you give us a little thought about that?
So for my research, the point of this is to try to identify those problem locations early on. And as you pointed out, like, no, we don't know what's causing those problems in the field, but you could, as a farmer, maybe go out to that specific area of the field instead of taking your time scouting the entire field frequently, you would identify that area that might be causing those issues and then be able to go out into the field and look at that specific area and determine what's causing the issue. So while my research isn't covering that, there is other research that's covering things of that same kind.
Yeah, so it's basically raising a red flag. Come out here and take a look at this area and see what might be going wrong. Yeah, thank you.
So Dorothy, Brian DeVries has a question. For the WIDIV part of your study, are you collecting flowering data? Do you think adjusting for flowering date would be useful?
So that's a great question, Brian, because we are collecting, I collected flowering data based on the images. So as I was taking each flight, I collected at what stage all of the plants were. And then there's also flowering data that's been collected previously on this data. And that will be used to make adjustments because early growth rate might just be predicting flowering date, as you pointed out. So.
Okay, we have a question from Ford Dennison. How does correlation between height and yield depend on plot size?
How does correlation between height and yield depend on plot size? I'm not sure I'm understanding the question. Do you mean, I don't know if I understand that question, but maybe they mean like as a bigger plot would have more consistent height.
No. Can you hear me?
Yeah, I can hear you.
So you would expect a positive correlation between height and yield if you have fairly small plots competing among each other or individual plants, right? You know, a tall plant next to a plot of short plants next to a plot of tall plants. The tall plants are going to have more yield because they're going to be shading the short plants. But does this correlation hold up as you get to field scale?
What do you mean, like production field scale for like the second objective, or do you mean
something big enough to avoid edge effects? So, you know, something with several meters on a side.
Right. So, something that we did do to try to take that into account is that these replications were randomized. So no plot would be next to the same plot from one replication to the next. So hopefully
that doesn't help at all.
No? Well, I guess that we don't have very big plots and we have single row plots. So that might be an issue that we need to consider. I know we had to throw out some plots.
Tony Fisher wrote about this back in the 60s or 70s, article called something like, are your yields messed up by competition among small plots or something like that. He's still alive, just spoke at the agronomy meetings. He's 90 something.
Okay. Yeah, I'll look into that. That's a good point. Since we only have single row plots, that could be a very big issue for this. So, thank you.
Okay, we have a question from Joanne Heron. In regards to your flights, have you seen any issues with the sunlight angle at different times or cloud cover?
Yes. So I did not take daytime or time of day or cloud cover into consideration as much when I was doing the flights. I tried to have it consistently cloudy or consistently sunny, I suppose, but not a whole lot. And that has had some issues with some of my data. On some of the days, there was one day in particular that I took my first flight too early in the morning, and that affected the accuracy of that data. And also there have been a couple of flights where there are clouds that are moving across the field as the flight is taking place. And most of the time that's not a huge issue with this kind of data, but there's one day in particular where there was a lot of difference between the resolution of those parts of the field, and that caused a lot of issues, and that day had to be completely thrown out.
Okay, we have a question from Amanda Waters. What is your recommendation for number of flights per season? How often would farmers need to capture data to get reasonable estimates for phenotypes? And are there plans to provide a GUI for farmers to process and view data easily?
So I'm not really sure what recommendations for number of flights per season is. I flew once a week, which resulted in those 10 flights from this year for the production field. And I think that seems pretty reasonable. It's very unrealistic to see farmers coming out more often than that, I think. And I think, yeah, that a once a week would probably be reasonable. And as we do more research, it'll be easier to tell how often that actually needs to be collected. And as of right now, there are not plans to provide a GUI for farmers to process and view the data easily on my part of the research. But as I said, the processing part of drone data is being tackled by a lot of different groups. And so as this data continues, there would probably be some working with those groups to try to provide a GUI to make this a lot easier for farmers to process.
Okay, great. Claire Menard has a question. She says, great talk. How transferable is this work to other crops?
So this would work pretty well for other crops. The direct models that we are producing, probably not, because growth rate in corn probably doesn't quite transfer to things like soybeans as well. But the initial work with trying to determine how those models can be formed can be transferable to trying to make those models in other crops.
Okay, let me just make sure, Dorothy, that I got everyone.
Okay.
Yeah, okay, so any last questions? Please type them now. One came right in. Michael Burns asks, how long can a drone stay in the air? Since the flight you showed was over a 2-acre field, if I'm remembering correctly, was this one continuous flight or did batteries need to be swapped? How would this translate to a field of roughly 100 acres?
So that's a great question, Michael. My drone can currently stay in the air for probably around 15 to 20 minutes. So that flight that I showed, as I said, was just over 2 acres, and it took a little over 17 minutes for that flight to be completed. But because of like getting into position and things like that, and the takeoff takes off more battery power than other things, I needed to change the battery usually near the very end of the flight. So this is another one of those issues that we covered in the cons of UAVs, that this doesn't translate well to a field of 100 acres. Because if you have to change the battery 50 times, that's not going to work at all. So hopefully as this data we continue collecting data, improvements in drones will continue being made that will make this a lot easier.
Okay. And Joanne Heron has one more question. Have you had issues with standability? How would you tackle problems such as green snap or root lodging?
So I personally have not had any issues with standability, but mostly because I only collected the data myself this year. And 2020 was great for standability. But I know of the previous 2 years with 2018 and 2019 data, there was a lot of lodging that occurred. And in those cases, when there was a wind event where there was a lot of lodging, we went out and collected flights for every day of recovery for the root lodging. I guess that's not as applicable for green snap because they're not going to recover. And in that case, we would probably just have to get rid of the plot that had green snap and probably the surrounding plots because of competition. This year, we also had some issues with drowned out plots in one of my fields, and those plots that were still standing were still removed because of the lack of competition. So.
Okay, I think that covers all of our questions.
This is Don. Let me ask one more.
Okay, go ahead, Don.
So within in the Wisconsin inbred materials, you have a wide range of genetic variation, right?
Yes.
What is the variation within the commercial hybrids? Is there a wide range of growth rates across the hybrids that you would be growing in a maturity zone?
I'm not entirely sure about the growth rates in all of the hybrids that might be grown in the maturity zone. For this project, we are flying it over one field with one hybrid in it.
Yeah.
And that hybrid, we're assuming, has different growth rates based on the health of the area where that plot is planted. Yeah. For instance, this data is from that production field. And you can see there is some difference in growth rate and plant height just based on the locations.
Yeah. So. I was just wondering, though, in terms of hybrid performance, when you're looking at growth rate, whether they were pretty similar in this day and age, or there was a big range in growth rates.
Yeah, I don't know that. I could look into that. That would be an interesting thing to look into.
Yeah, I can Google it too, probably. Thank you.
Mm-hmm.
Dorothy, we had another question coming in from Brian DeVries. Staging of commercial hybrids for farmers is very important. What ways could UAVs be used to study that in relation to final yield?
So my project isn't really focusing on that, but that would be very interesting. I know that some research has been done on plot color over time. And that could be done easily by having set color grids out in the field. And that might have an interesting impact on yield, as it probably has a higher yield the longer the plant stays green. But I haven't looked into that at all.
Okay. If there are any other further questions, now is the time to type them into the Q&A or the chat. I don't see any more coming in. So thank you everyone for attending, and thank you, Dorothy, for that great talk.